Okay, so today we're going to be looking at the lac operon in more detail. Um, Complex, but as long as you remember the names of the different parts, it actually makes logical sense. So here, this is our piece of DNA where we actually have three separate genes here that make up the enzyme lactase. So these are the three genes that code for the enzyme lactase. And lactase is an enzyme that breaks down the sugar in milk into two monosaccharides, okay, so that it can be digested. So if you're lactose intolerant, you are missing this enzyme. So you are not making this enzyme. So there's something going on with your gene regulation of this enzyme. Typically, people stopped making this enzyme when they were about 3 years old because back in the day, people only drank milk from their mothers. Um, then people started drinking milk from other organisms like cows, and a mutation occurred which kept this gene being expressed. And so people who are lactose intolerant, that's actually, they don't have the mutation. The people who can continue to drink milk and break down lactose beyond 3 years old actually are the ones with a mutation. But our control of this gene is way more complex than this. So this is a very simple way of controlling gene expression, and it's done in things like bacteria. So hopefully you remember we have a promoter. So that's the section so that RNA polymerase knows there's genes here. I need to make a messenger RNA copy. I'm going to attach to the promoter. And there's also this little red piece is called the operator. Okay? And this green thing here is called the repressor. Now, the repressor does exactly what a repressor should do. It stops. It stops the transcription of this set of genes. So it represses transcription or making the messenger RNA copy. And this yellow piece here is, of course, RNA polymerase. Okay. So what is going on here is when there is no lactose, so if there is no lactose, which is the sugar we need to break down, present, then we have this repressor, then the repressor binds to the operator. So you can see there's a lot of language here, right? That you have to just get your head around. So the repressor binds to the operator, oops, preventing transcription. So literally RNA polymerase is blocked. So RNA is blocked. Can't get through there. Just cannot transcribe at this point. However, as soon as lactose enters into the system, then something different happens. So here, these represent lactose. So if we're looking at a bacteria, it pulled some lactose in, it pulled some of this sugar in. And now it's inside the cell, and the lactose will bind to the repressor. You can see there's lactose right here binding to the repressor. That changes the shape of the repressor, and the repressor is now removed from the operator, allowing RNA polymerase to go through and transcribe and then eventually translate. So when lactose is present, it binds to the repressor, changing its shape. It releases from the operator, allowing transcription. Okay, and then ultimately what we have to realize is if these three genes are allowed to undergo transcription and translation, they will produce an enzyme. That enzyme is going to be now in the cell, in the system, and the enzyme is going to come around and it's going to break down all this lactose. So as the enzyme breaks down the lactose, it will get rid of the lactose in the system, including the one that's bound in the repressor. So when all of the lactose is broken down, including the one in the repressor will come back down and bind again to the operator and will prevent transcription and translation. Why? Because we don't need it anymore. All the lactose is broken down. So hopefully that system makes sense to you. It is pretty logical. So I also am going to add a link to a simulation. It's called PhET. You may need to download it to your computer, but then you can just get rid of it after. Try and work through the simulation and see if you can make it work, because it's a simulation all about this lac operon, and it really helps you understand what is going on. So, so once all the lactose is broken down, including the one in the repressor, the repressor will bind to the operator again, preventing transcription. So hopefully you understand that, but that little simulation should really help. So anyway, you have this picture, so you just want to, take down the parts, write a little note on how it works. Yeah, we don't. Hopefully, I can get that. And then just one last thing here. If you could take down this note, gene control mechanisms. I know you looked at there's pre-transcription, transcriptional, translational ways of controlling genes in eukaryotes. That's us. Very complex. The lac operon is as simple as it gets, and that doesn't apply to us. So this is just a really sort of important way we control whether we are expressing genes or not. And it deals with functional groups. So acetyl groups, which are these right here, are, if you remember, our DNA has histones, which are proteins, and that's what the DNA wraps itself around to keep itself untangled and organized. Acetyl groups are added to those histones, and what that does is it loosens the association of the DNA with the histones, or in essence, unwraps them from the histones and makes them accessible for transcription. So genes that you use all the time, there are a lot of acetyl groups attached to the histones. okay, that they're wrapped around, because we want them sort of available for transcription and translation all the time. So this is how your environment sort of, not sort of, it's how your environment triggers expression. The more you use a gene, the more available it will be for use. Okay, so you can, through your environment, make genes very accessible for expression over time, over your lifetime, right? So that's how environment affects genetic expression. And we see that in identical twins, because when identical twins are born, they look identical. But as they age, they look less and less alike. So, what you're seeing is the environment affecting the expression of their genes, because their genes are identical. So, the reason they look different is environmental. So, we know both environment and genes come into play with who a person is, but one influences the other. So the more complex the trait, things like intelligence and personality, your environment can really affect those genes and how they're expressed. Um, and then conversely, we can shut down genes as well so that they're not expressed. And again, that's a functional group. Um, and it's called methylation. So these methyl groups are added to the cytosine bases in the promoter of a gene, and they inhibit transcription. We call that silencing. Okay, so genes can be silenced. And some genes can be silenced from one generation to the other. Some genes are permanently silenced. Our tail, I keep talking about the tail genes because it's so interesting, are genes that we have that would, should they be turned on, would have given us a tail when we were born. They are silenced through methyl groups, and that is passed from one generation to another. So what they're finding is this process of methylation or acetylation for some genes can be passed from one generation to the other, giving genetics a whole other level of complexity. What that means is your actions in your lifetime can, in fact, silence or make available certain genes more or less for your offspring. And that is called epigenetics. And it's very new in genetics and very interesting, because we always assumed that people were born with a clean slate. You have your genes and you inherited those, but the environment your parents were in and how that affected the expression of those genes did not correlate with the offspring, or in essence, they were wiped clean. All the methylizations and acetylizations that were accumulated over their lifetime were wiped clean. And what we're finding is that they're actually not for certain genes. So you can actually affect your own children's genetic expression through what you do to yourself in your lifetime. So that is the study of epigenetics, and you just want to take down what epigenetics is. Really, really interesting stuff. And very complex, but it's opening up a whole new world of genetics to study.